I'm Daniel Levine, and this is the Bio Report. Elise, thanks for joining us.
Thank you, Daniel.
We're going to talk about biotechnology, generative AI, and Cradle's efforts to use its technology to accelerate the development of proteins for not only therapeutic applications, but a broad range of bioproducts. We focus largely on human therapeutics on the show, but proteins are proteins. Before we talk about what Cradle's doing, can you start with the larger bioeconomy and the potential for biotechnology to transform industrial production more broadly?
Yeah, for sure. I mean, these are some topics that are very, very close to my heart. I truly believe that biotechnology has the potential to become one of the biggest drivers of societal transformation. We're obviously faced globally with some pretty significant challenges. We need to deliver improved health outcomes for a world population that is aging. We also need more sustainable ways to produce products that are critical to us while reducing their environmental burden. And if you think about it, a lot of the material that we use around us comes from biology. I mean, we eat plants, we eat animals, we use wood for construction and fuel and paper. But there's so many ways in which more technologically advanced products inspired or coming from biology can show up in our lives. And this is, of course, in therapeutics, like antibodies that you might use if you're faced with cancer, or the COVID vaccine coming from mRNA or from protein. But on the other use cases, you also think of, for example, the enzymes that help wash your clothes at low temperatures. And these, this sounds like maybe a mundane application, but if you think about removing fat and oil and protein stains at low temperature, and then consider that the use of these enzymes in washing powder can eliminate 1,600 pounds of carbon dioxide emissions per household per year. And think about the fact that after the introduction of these enzymes, the energy consumption that was used in clothes washers actually dropped by 75% in the 2 decades that followed the introduction. There are really ways in which, biotechnology products can affect us. Yeah, I think for the better. And of course, the challenge is rather than growing a plant and eating it directly, oftentimes these products are inspired by nature or have a starting point in nature, but they have to be engineered further to actually become commercially viable and useful for the application that we as humans have in mind for them. And this is something that can take a long time and be quite expensive. When you're talking about protein design, there's often trial and error methods that are involved. Many of the designs that you test along the way fail to meet their specifications. And this is where we can start to now bring in these new technologies like generative AI to really accelerate that process of bringing these new bio-based products to market.
You did start your career on the industrial side of biotechnology, having worked at Zymergen, and before that, working to engineer animal-free dairy proteins. What's the case for enabling bio-based production, and why should more traditional manufacturers be thinking about transforming their processes to enable themselves to use this technology?
Well, if you think about it, bio-based production often occurs in much milder conditions, and it often has less damaging waste products compared to, for example, chemical synthesis. If you think about it, if a cell produces a lot of toxin, it ends up killing itself, and so it's not a very productive system to use. And compared to traditional methods, bio-based production can offer alternative production methods that end up saving a lot of energy or land or water. This is the case, for example, in substituting proteins from cows for proteins from microbial fermentation, like Perfect Day is doing. But also moving forward, it can help to tackle resource scarcity, because petrochemicals may not be always as abundantly and as cheaply available as we take them for granted today. And this is where bio-based production processes can substitute and create some of the stuff that we have around us. But also things like how to unlock precious metals from mining ores in ways that conventional mining may not be able to do. This is where biotech can offer ways to use microorganisms to do just that. So if we're able to just transition a fraction of the existing methods to bio-based solutions, we can really have already a very positive impact on the planet. Now, of course, this is in, for example, the industrial or personal care or food space. The same also applies if you're thinking about novel therapeutic proteins, not only their efficacy, but also how easy is it to manufacture them and how sustainably can they be shipped around the world to reach the places where they're needed most.
Cradle's working with customers across the biotechnology spectrum in both therapeutics and industrial applications. While there may be issues around such things as economics and scale and purity, is there a difference in designing a protein for a therapeutic rather than an industrial application?
There is. I mean, depending on the application, the protein will have very different requirements for the final application. So if you think about research and discovery for therapeutics, that will often start by defining this target product profile, which involves defining what the protein, for example, in an antibody or a vaccine, how efficacious it must be, what are the conditions to use it safely in a human body, and how does it meet manufacturability requirements. And if your therapeutic protein just fails to meet one of these criteria, it won't make it onto the market. It won't pass clinical trials, or you won't be able to produce enough of it. So there's a really long way to go between identifying that initial hit or a promising candidate and really developing it into a lead that makes it all the way into clinical trials and beyond. Whereas for diagnostics or maybe biocatalysis or industrial applications, the main requirement will not be so much the safety of a protein inside the human body and how it responds to this, but maybe more stringent on activity, affinity, scalability, manufacturability. And with this list of final requirements, basically the challenge that R&D scientists are faced with is, can we develop an assay to test for the required behavior, but at the lab scale? And can the scientist introduce mutations to improve the protein so that it starts to meet this property and do that for each property without destroying all the other ones that are also important? And the beauty of this challenge, which is so different depending on the protein and the specific application, is that given sufficient amount of lab data, these properties are learnable. They're learnable for humans sometimes, but they're also learnable for machine learning algorithms in a way that is sometimes much more efficient and can really cut out time to get that product to market.
In the realm of AI applied to proteins, listeners may have heard of AlphaFold, which is an AI system used to predict the three-dimensional structure of proteins. Cradle's doing something fundamentally different. Can you explain what your AI system does?
Of course. So if you think about AlphaFold, it uses deep learning to predict the shape of a protein, a structure of a protein in 3 dimensions. And this is a predictive algorithm because given a protein sequence, so the string of amino acid, it predicts the 3D structure. Cradle is different because we have built a platform for protein design using generative machine learning. Now, what do I mean by the generative machine learning? If you think about ChatGPT, which is a generative model, it generates natural language or human sequences, and it's trained on the internet, so human language. And given a query, like a question, it can generate an answer for you. Now, that answer may or may not be correct. That's for you to do the fact checking. And similarly, Cradle's models generate new protein sequences, so sequences that contain certain amino acid mutations that are meant to improve the property that you care about for your final application. And so the prompt wouldn't be a question, but would be the sequence that you provide at your starting point and an objective, such as improving the binding affinity to an epitope or increasing the thermostability of your molecule. And then what Cradle's generative machine learning does through this platform is propose a set of sequences, so a small library that you can evaluate in the wet lab. Now, of course, these sequences aren't generated randomly, and if ChatGPT hallucinates and comes up with a bogus answer, then it's only a quick Google search to verify that. Whereas if you're sent into the lab with a sequence that makes no sense or is never going to work, that's going to be a huge waste of time and money. And so the whole point of using Cradle and these machine learning methods is to go into the lab with designs that are of better quality and have a higher likelihood of meeting your design objectives. And so ultimately getting to your goal in fewer experimental rounds. And in order to do that effectively and to be a useful product to people, of course, this machine learning has to be better than what a human could come up with rational design approaches or more traditional trial and error screening approaches.
Well, help us understand that. I'm sure most listeners by now have had the experience of writing a prompt for a consumer-facing AI engine, but what does it take to get your system to generate a specific protein for a specific task? What does it take to write a prompt, or what other inputs do you need to provide?
ChatGPT was trained on the internet, so that's human or natural language, whereas the models that Cradle uses were trained on protein language, so protein sequences, the strings of amino acids. And instead of asking a text prompt question, you actually give the model a starting sequence, and then you ask it to improve for a specific property. So you inform when you start a project which properties you care about and how you're going to measure them and what you expect the value of that measurement to do. Do you want it to go up? Do you want it to go down? Does it need to meet a certain threshold for that candidate to be good in your objective? And instead of a text answer, the models will take this sequence and then use all their base knowledge of the protein language, as well as any evolutionary trajectory of the sequence that you fed in, as well as any prior data that you fed in. And what they will do is the answer that they will generate is a set of sequences, so a small library that you can assay and test in your own wet lab. And the idea is that this small library, each of those sequences has not only a higher likelihood of meeting your design objectives, so be it better binding affinity or higher stability. These sequences are also very diverse, so that you have multiple shots on goal, and there's really learning that can happen over multiple rounds. What we also see is that now with these large machine learning approaches, the data set that you generate in each round can be limited to just about a microtiter plate, so just about 96 proteins tested per round. This is something that more and more labs these days are able to do. And so by testing the answer, so the set of sequences in your lab, and feeding those results back into the machine learning model, you start to get this loop where the model, from a generic protein model that knows something about stability, that knows about the evolutionary trajectory of proteins, becomes really an expert on the project and the task that you're trying to solve across all these different protein properties. And that's where the magic happens.
And I know in the case of generative AI, when applied to small molecule drugs, if you get it to enhance one property, it's often at the expense of another. Is that the case with protein design?
That can absolutely happen. Some protein properties evolve kind of hand in hand. They strengthen each other. Some can be opposing. And this is a really great challenge for machine learning, and one that is very hard for the human brain, is to make these trade-offs. And so when you use Cradle for a multi-property optimization problem, the models will be able to learn those trade-offs and then generate sequences that really are meaningful to you and where you're trying to get for that final case. And of course, sometimes nature says no, right? In some cases, a project may be hard to solve because you run into physical constraints, like the limits of physics. So for enzymatic activity, we have had a case in which an enzyme was already pretty pretty evolved through multiple rounds of mutagenesis. And the models had such a hard time finding sequences that they thought would be better that we actually ended up advising against running that next experimental round because the chances of success were so low. And so not only can machine learning help to produce better designs, it can also help you to inform when maybe the time is right to stop and change your search strategy or your approach, which again saves time and money in the wet lab.
Does the system find proteins that meet a user's criteria, or does it begin with a known protein and then optimize it, or does it actually create proteins that might not exist in nature?
Well, nature offers a great starting point for discovery. Many of the drugs, small molecule and large molecule, that we use today are actually inspired from natural starting points. But almost by definition, in this type of biotechnology, you are taking a protein from its natural context and placing it in a very different context. So subjecting it to all these requirements that need to be met in order for it to be valuable to you as a human drug developer or industrial biotechnologist. And so starting from an initial starting point, this could be a natural sequence or maybe a weak binder that came out of an immunization campaign if you're working on therapeutics. Then the magic happens when the models generate a set of variants on that starting sequence or a starting set of sequences. And for you to go into the wet lab, generate data such that the models can learn from the specific project data and really help to move you from that starting point round over round to your final objective. So from a weak binder, from an immunization campaign, to a high affinity binder that also shows, great T cell activation and also has low immunogenicity and high stability. And so learning really happens in a loop. This is something that we have focused on a lot at Cradle. We think that in the industry, there's a lot of attention to what is known as zero-shot models. So models that are able to predict a first set of designs, go test these in the lab. But what happens after? Like, how do you take the next step? And there's a lot of nuance in building this loop so that the machine learning can use all its knowledge from these large base models that have seen all publicly available protein sequences to becoming experts at your specific project. I can say a little bit more about that.
Yeah, I actually want
to loop, but
I want to go a little deeper on two of those points. Great. First is, how much of this takes place in silico? At what point does someone take what the system has generated and then test it in a wet lab?
So you will need in silico and wet lab testing. Any ML company claiming to replace wet lab work in the field of protein design is selling snake oil. Luckily, we haven't come across too many of them. But the whole idea of the Cradle platform is that you leverage generative ML as a design partner while also conducting experiments in the wet lab. So making more efficient use of the capacity in the wet lab. So for computational users, this means you can bring your own models to the Cradle platform and connect them to the workflow. For experimental users, you can really collaborate with generative machine learning models by evaluating their designs and combining your own rational designs with those from Cradle's ML. But to say a little bit more about that learning loop and how the interplay between in silico and wet lab works, is that we start from these base models that have been trained on all known protein sequences. And this allows the generator, which we can envision as the painter, to propose a new set of sequences. So if you think about the generator as the painter, it will paint 500,000 paintings, where the objective maybe is to make something that looks like it was painted by Rembrandt, right? The predictor then comes in as the art critic that scores these sequences. So for every painting, it will look at it and go, how much does this look like a Rembrandt, and score that from 0 to 1 or from 1 to 10. It will score these paintings. And then we have a smart way to basically select the top pieces. So you can't bring 500,000 paintings to an art auction, but maybe you just want to bring 10 or 100. And with these down selected sequences, you can then go to the art auction or move into the wet lab. And the important thing is to close that learning loop. So to then take the output that's coming out of the wet lab, so the real world measured value, and show those again to the generator and the predictor, such that both learn how to suggest better sequences, so sequences that are more likely to succeed, and to be better able to differentiate between those sequences that are a little bit better in their performance versus those that are a little bit worse. And so the painter becomes better at painting things that look like Rembrandt, and the predictor becomes a better art critic to differentiate the Rembrandts from the not so Rembrandts. And so in this way, you can move from a natural starting sequence into much more novel sequences that fulfill all the requirements for your target product profile.
And to what extent are your customers using their own data sets, or are they using some Cradle data set?
A really important part of how we've built Cradle is that we have a large team of ML researchers and engineers, but we also have an in-house wet lab. And so we generate our internal data sets with the objective to make the best learning system. So rather than trying to make one model to rule them all, so one giant data set to feed the world's best thermostability machine learning model, we're actually simulating real world experiments in our own lab by having many different projects and many different objectives, and seeing how good the Cradle core infrastructure is at learning those properties and moving those projects towards their final objectives.
And has Cradle relied only on public data sets, or has it generated, unique data sets to train the system? And what has it had to do to validate its system?
Yeah, so we use a combination of public sequence data as well as proprietary experimental data, and always aim to make the most of the experimental data that is generated in a specific project by our customer. Now, of course, customers can choose to run these experiments in their own wet lab or outsource them to CRO partners. If you are interested in using Cradle's platform, but you don't have wet lab capacity, we can always connect you to a CRO partner. One example is a partnership that we have with Ginkgo Bioworks, who can run experiments on demand in their biofoundry for both enzymes as well as antibodies. Together the combination of Cradle's core infrastructure, so the public and the proprietary data that went into those core models, with that training that our models have had to become really, really good learning systems, plus the data that you as a customer collect on your project, that's really what all adds up to decreasing the time that you spend in that optimization phase. And of course, for biopharma, lead optimization is where the majority of the money and the time is spent. And so decreasing the time spent in that lead optimization phase reduces the cost of development, and also gets that product filed at IND faster and onto the market faster, which is hugely valuable. So we've shown within projects that we've run with partners like Johnson & Johnson Innovative Medicine and Twist Biosciences and Novonesis, that we can really speed up this lead optimization phase anywhere between 2.5 and 12 times. What you can do while you're testing or building these models is continuously run benchmarks to compare the performance of Cradle's models to the performance of other models out there, and really test how they perform on very different data sets. The challenge, of course, is that there aren't very many multi-round, multi-mutant data sets out there in the public domain. So that's something that we invest in building in-house to really make sure that as we continue to onboard new features and support new types of protein challenges, that our generative machine learning system continues to be the state of the art.
The system continuously learns from what your customers are doing, but are those learnings unique to the customer themselves, or do they somehow feed back into the broader system?
So data security and privacy is paramount. The company, like Cradle, would not exist if we weren't able to guarantee that any data that a customer uploads to the platform is secure and private to them. So we never ever mix data between customers. We also never mix or expose models that have interacted with customer data between customers. So if you think of a balloon factory, like we supply the balloon, so that the machine learning model, and the customer supplies the air that fills the balloon. After use, the balloon is popped, or you get to take it home or give it to your kids or do what you want with it, but it will never be given to the next party goer. And so then the follow-up question that I could feel coming is, so how do we get better, right? And that's really where we focus at Cradle on making our models the best learning system possible, using our wet lab, like I said, kind of like a racetrack. So testing very different enzymes and antibodies and proteins for different properties and evaluating how our machine learning can best learn to optimize different properties. That is what you get access to as a user. So a very easy way to use these models that are really the best at learning very quickly about a specific protein problem. We've used the analogy, if you're building a self-driving car, you have to have a racetrack to race that on. So we need a wet lab to test our models on, and preferably if you're testing the self-driving car, you're not just driving it around one block repeatedly, right? Which is what you kind of end up doing if you're a company with a limited internal data set. Rather, you'd much rather have many more examples of neighborhoods and traffic situations to make sure that that model is better able to accommodate all the different things that society can muster up on the roads.
And can anything be said yet about how the system can save a customer either time or money in engineering a protein relative to more traditional approaches?
Absolutely. So with every project, we try and evaluate together with our customer how much faster, how much more efficient was it using machine learning compared to just traditional approaches. We've seen that range between 2.5 to 12 times, like I said, compared to industry benchmarks. And I mean, the cost savings there can be very significant. If you think about, according to the research in the Journal of Healthcare Economics, the average investment in the R&D to develop a single drug ranges from 280 to 380 million, with the preclinical costs accounting for about 43% of that. So anything that you can do to cut out that an experimental cycle or multiple cycles, and get the drug into the preclinical phase is such a gain for biopharma.
How do you work with customers? What's the business model? Is this software as a service? Do you work on a project basis, or do you enter collaborations with partners and have potential upside in an end product?
So we operate as a SaaS model, so software as a service on specific projects with our customers. The companies that use our platform fully own their data. They also own the IP rights from any sequence that they develop and test using Cradle. And we don't take royalty or milestone payments. We're really a tool that can accelerate protein design. And we see that this is valuable to industry leaders across different sectors, so pharmaceuticals, chemicals, food and materials. I've named a couple like Johnson & Johnson, Innovative Medicine, Novonesis, Twist Biosciences, etc. We really believe that there is such a huge potential for generative machine learning to accelerate protein design and accelerate biotechnology more broadly. And we think that impact, the fastest way to reach that is really by making sure that every lab can have access to the state-of-the-art AI methods. That's why we started this company in October 2021. That's still the mission that we're on, and it's really fun to be able to work with so many teams across the industry working on such important research using the tools that we've developed at Cradle. And maybe, oh, sorry. Well, one thing that I didn't call out, which maybe now is a good time to do, is that it's a software platform, right? You can use generative machine learning. I've called out that computational users can bring their own models, experimental users can interact with it, but really the thing that allows this usability of the platform is that we invest in user experience research. So similar to all the apps that you may use in your daily life, whether you're booking a taxi at Uber or getting a hotel for your vacation or whatnot, all of these platforms are designed by talking to a lot of users, understanding how they would like to interact with the software, making sure that the most obvious questions are answered. This is something that we invest in as well to make sure that the platform is accessible, whether you are a research associate performing the wet lab experiments or a project manager curious to see the progression of a project, or a bioinformatician or a machine learning practitioner. So it's really, with this innovation comes a huge amount of change management in these organizations. And we see Cradle as a platform where all these different types of users with their different interests can come together and collaborate on protein design to bring those products to market faster.
You've talked about using the system to engineer specific qualities into a protein. I'm wondering, are there specific things beyond a basic function of a protein that you find customers try to engineer into their end product?
Gosh, yeah, there's as many people, as many ideas as there are protein projects to come up with. So in biologics, binding affinity, efficacy, immunogenicity, vaccines, maybe thinking about equitable access. So how can you ship it at not cold chain across the world, increasing stability. But there's also peptides that maybe need to get into a cell, so cell penetration. For biocatalysis on the enzyme side, activity, but also specificity, stability, maybe the ability to work on novel, like new substrates or produce new reactions. There's really a plethora of things that people are trying to do using proteins.
Is it common to see them engineer proteins to make them more manufacturable or scalable?
That definitely comes up in the process. So once you have a protein that carries out a particular function or binds to your liking, then the way for that to scale into the market is to be able to make more of it. Now, of course, that's a greater challenge for the high volume, low value products, so the bulk products, as you might see in industrial applications. Maybe more so than a therapeutic for a rare disease, of which only a few doses are needed globally. But regardless of the process, manufacturability and scalability do come up as targets. Yeah.
Cradle raised $24 million in a Series A funding round in 2023. It's raised a total of $33 million to date. How far will existing funding take you, and what's the plan for raising additional capital?
So we have utilized the raised capital for expanding our AI and ML research, building out new features on the platform, carrying out our internal wet lab research, amongst other things. So we have large ambitions, and we've been lucky to get backing from great investors like Index Ventures and Kindred Capital to really help us on this journey and moving towards this impact. So I think the sky is the limit for these methods, and I really hope and believe that Cradle can play a big role in this new development of bringing generative AI into protein design workflows. Yeah. We, in terms of like what the future holds, we're always speaking with investors, but don't have anything more to share on that right now.
Elise de Reus, co-founder of Cradle. Elise, thanks so much for your time today.
Thank you, Denny. It was a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, Subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.